Hello students and welcome back to Microbial Concepts. So today's video is regarding getting familiar with microbiological terms, and this is second part. So already a first part is uploaded on my channel. You can watch that out, and this video is actually for freshers or the newbies to microbiology, or even non-microbiologists who want to learn about some of these terms. They can get their concepts clear. Okay, so let's start. So first term is staining. Now staining is one of the techniques that we use while we are studying some specimens, say bacterial or algal or fungal specimens in case of microbiology, by mounting them. Now mounting is a technique where we take a small sample or a small part of our specimen and we place it on our glass slide and we try to observe it under microscope. Now the specimen will, mostly the bacterial specimens, they don't have any kind of color, so you will observe them, or you will see that those appear transparent. Now to add some contrast to our specimen, we need some dyes and stains that we use, and they make it easier for us to observe and study and differentiate between the bacteria. Okay, so not just bacteria, even algal or not algal, the fungal structures. Okay, we don't stay in the algal structures. They already have their own color and we observe them directly under microscope. Okay, so in case of bacteria and fungi, we need the staining techniques. Okay, so these are some staining techniques which I have listed here. So you can see the first one is gram staining, which you are taught when you first appear to the first year. Okay, very first technique that is taught to you under microbiology syllabus. Okay, so to differentiate bacteria depending on their cell wall composition, we do gram staining. This is gram negative bacteria and this picture is of a gram positive bacteria. Then endospore staining. So endospore staining is of this. Okay, their endospore is a part of bacteria, which bacteria they produce endospores when the unfavorable conditions are present. Okay, the cell don't want to die, so it gets converted, or it produces it endospore. Okay, and it gets itself converted into a dormant cell. Now, what is a dormant cell? That I will discuss in the further slide. Now next is flagella staining, acid fast staining. So this is acid fast staining slide. Then monochrome staining is where any one kind of dye is used to stain the specimen. Okay, you don't differentiate between these specimens, but you can just observe the cell size and cell shape. Okay, for that purpose we use monochrome staining. Then lactophenol cotton blue staining is actually for fungi. So you can see here, this is aspergillus, which is stained by using lactophenol cotton blue staining, and it helps us to study the structure, the spore arrangement, and it helps in fungal identification. Okay, so this is about staining. Now next is about spores and endospores. So spores, we have seen that this is a structure of a fungi, that is an aspergillus. So you can see there is a sporal arrangement. Okay, there are long thread like structures which where all the spores are arranged. Okay, so the spores of fungi, they are actually reproductive. That means a single spore of this aspergillus, if it lands on a substrate where all the required favorable conditions are present, then a single spore can germinate and can grow into a full mature structure of a fungi, that is aspergillus here. So it serves a function analogous to that of seed. That means same as the seed has function in plants. That means when seed gets all the required conditions like sunlight, water, nutrients from the soil, it germinates and it grows into a plant. Same function is served by the spores of fungi. So they are produced and released by the fruiting bodies of fungi. So this is a fruiting body of aspergillus which is terminally present, and the fruiting body has these spores. So when the spores get favorable condition, it germinates and grows into a full mature structure of fungi. The spore arrangement is used in the fungal identification. Now in case of endospores, now spores we have studied, they are produced by fungi. Endospores are produced by bacteria only. So they are small, oval and spherical and are highly resistant to high temperatures, chemicals, desiccation and radiation. That means this endospore is highly resistant to all the adverse conditions. Okay, desiccation is where very or extreme dry conditions are present, there is no moisture, no water. Okay, so under unfavorable conditions, bacteria they undergo sporulation. Now this kind of sporulation is what? Endospore formation. Okay, Endospore formation is also termed as sporulation when you do long fermentations where a batch is taken of bacillus. Say for example bacillus produces endospore, you will learn that. So you say that okay, now the bacteria is going sporulation, it is undergoing sporulation. That means it is getting itself, the vegetative cells are getting themselves converted into a endospore. Okay, now it allows bacteria to produce a dormant cell that can preserve the genetic material. The main purpose is to preserve the genetic material, and the bacteria can grow back when the favorable conditions are present. Now what is dormant cell? Dormant cell is cell, or the endospore is actually a dormant cell which can live for very long period of time, even for hundreds of years a dormant endospore cell can live. Okay, then comes flagella. Flagella is a hair-like appendage that protrudes from a bacterial cell. So you can see here a thread-like structure that comes out of a bacterial cell is actually a flagella. So it is actually a motility organ that enables motility, or you can say movement, and chemotaxis. Now bacteria, they can have one flagella or no flagella and several flagella. So there are different types even depending on the number of flagella and the position of flagella. Bacteria, they are classified, okay, or they are even identified. So Atricus is one which don't have any kind of flagella. Example is Micrococcus. Monotrichus is Vibrio, which has one flagella. Amphitricus is Nitrosomonas, which has two flagellas on the terminal side either. And then Lophotrichus is Salmonella, which has at least 3 to 4 flagella on the terminal sides. Okay. Then Peritrichus is many flagella. It has many flagella like E. coli. And Cephalotrichus is Corynebacterium, which has group of flagella on a one side of one terminal side. Okay, so depending on the position of flagella and number of flagella, you can identify and differentiate between your bacterial species. Now capsule. So capsule is one of the cell organelle, and it is the outermost structure which is found in most of the bacteria and fungal cell. Now the function is to protect microbial cells from immune recognition. That means when in case of pathogens, these capsules, they play a very important role, as the pathogen has a role to, or it has main role to invade a human body, and it has to infect the human body. But the immune system of human body will start to recognize, and it will start killing the bacterial cell. But the capsule which is present towards the outer side of a microbial cell, it will resist the immune system. How? It will protect from immune recognition. That means the host body cannot recognize that yes, this one is the pathogen which is causing the infection, and it will prevent from killing it. Now it is made up of a repeating polysaccharides. Now the capsule is how it is made up of a repeating polysaccharides, and it is located outside the cell membrane. And in case of gram-negative bacteria, it covers the lipopolysaccharide. Okay, it is towards the outer side of the lipopolysaccharide. So this is about flagella and capsule. Then genus, species, and strain. Now all these are taxonomic ranks. Now we have studied different taxonomic ranks, like say first is domain, then kingdom, phyla, class, order, family, and then after family comes the genus, species, and strain. Okay, so we have studied this kind of taxonomic ranking, say from our secondary grades. So the further comes the genus, species, and strain, and in case of microbiology, you mostly deal with these. Okay, these taxonomic ranks. So first is genus. A genus is a taxonomic rank which comes after family. Okay, so it is a group of phylogenetically related living things and is made up of one or more species. The plural form is genera and singular form is genus. Okay, so you can see here the lactobacillus is the first name of the bacteria that we write is the genus, and the first letter is capital. In case of species that we will study now, it is the second name and the first letter is small. Okay, and we write it in the italics. Now the species. Species is a taxonomic rank which comes after genus, and after species comes the strain. So a group of living organisms consisting of similar individuals which are capable of exchanging genes and interbreeding is known as species. And a strain is a genetic variant or a subtype within a biological species. Okay, so a subtype within this particular species is known as strain. So Lactobacillus acidophilus can have different strains. Okay, so this is the name of a particular bacteria till species level. After species level, even there is identification done, and that is of this subtype of the species, which is known as strain. Okay, so this is about genus, species, and strain. Now gene and chromosome. Now many students they get confused between these terms, so get it clarified here. So this is the cell has nucleus has chromosomes. Okay, now the chromosome is the main structure which has our DNA covered, and the DNA has different genes which we study. Okay, so what is gene? Gene is a segment of DNA. A single or a small part of a DNA is known as gene that contains the code for a specific protein. And what is chromosome? Chromosome is a structure within a cell that contains the person's gene. Now different or too many genes, you can say, they make up a DNA, and this particular DNA is what is uh, chromosome is having on it. Okay, so chromosome is a structure within a cell that contains the person's gene or DNA. Now fermentation. A best example to give of fermentation is we ferment our batter of idli, or say um, other other food items where we do fermentation to get a specific taste and texture. Okay, so fermentation is a biochemical process that gets energy from carbohydrates and does not require oxygen. Now how this fermentation takes place? Now there are some bacteria which require no oxygen to use carbohydrates. Okay, and they produce some byproducts, or they give some byproducts like sugars and starch are converted into alcohol, carbon dioxide, and acid. Okay, so these organisms they work under no oxygen, or they don't require any oxygen, and they can be anaerobic or facultatively anaerobic, and they can work under such environments and give out some products like carbon dioxide, alcohol, and acid, organic acid. Now that depends on the type of fermentation and type of substrate or type of carbohydrate that is being used. Okay, so best example to write, if you have this in your first year, you have a short note, then you can write these are the examples. Now the byproduct is used in bread making. That means the carbon dioxide which is released while bread making, it gives the spongy texture to our bread. Then in case of brewery, where ethanol and alcohols they are produced, the ethanol is the or the alcohol is the byproduct that is used here. In case of lactic acid, that is the organic acid produced by while fermentation, that gives the distinctive taste to cheese. Okay, so the cheese industry also use fermentation, and the lactic acid is the um, uh, main substance here that gives a distinctive taste to your cheese. Then comes the pasteurization. So this is the process of heating every particle of milk or milk product in a properly designed and operated equipment at specified time and temperature. It kills harmful microbes in milk without affecting the taste and nutritional value. So what happens? It increases the shelf life of milk, and it helps in storage and long transport of milk. So this is about the pasteurization. I am not going in details here, but if you want to know more, you can watch my videos which are uploaded on my channel, that is why milk is pasteurized, and a detailed video on pasteurization with the types of pasteurization, everything explained. Okay, then comes the immunity. Now there is separate subtopic under microbiology that you will deal further is immunology. Now immunology is study of immune system of humans. Now we have a immune system, that means the organs like thymus, bone marrow, lymph nodes, and cells like white blood cells, and some processes that helps us fight infection. So that is immunity that we get from our some organs and some cells which are present in our body. Now, immunity is a complex biological system endowed with the capacity to recognize and tolerate whatever belongs to self and to recognize and reject whatever is foreign. That means anything that is not self, the immune system or the immune cells will recognize them as foreign and they will start killing them. Okay, so that's the immunity, and immunology is the subject where you deal everything that is related to the immunity of humans. Okay, now antigens and antibody is also a part of immunity. So what is antigen and antibody? So antigen is any kind of substance that can induce immune response. Now immune response is what, uh, when we get infected, what happens? The first symptom that is seen is fever. Okay, so why there is fever? Because our immune system is getting triggered and it is fighting with the infection. So that is the immune response. Okay, so you can see here this structure, y-shaped structure, is of antibody, and these are the antigens. So antigens, they can bind to or antibodies, they can actually bind to antigens and they can recognize and they can trigger the immune system. Okay, now this is actually a lock and key type of a model. That means each and every antibody is specific for each and every antigen. Okay, so that is the difference between antigen and antibody. So antigen is a foreign substance and antibody is our own um, immune immune system structure that our own body it produces. Now an antibody is also known as immunoglobulin. It is a large Y-shaped protein used by immune system to identify and neutralize foreign bodies in host body. Now the function is specific binding to the antigens and activation of other components of immune system to neutralize them. Okay, that's the role here. So when antigen comes into the picture, you will find antibodies fighting against them. Okay, then comes disease and infection. So disease, it is defined as a disorder of structure or a function of human, animal or plant. It is identified by symptoms produced. Okay, now infection is what? Now there is difference between disease and infection. So infection is invasion and growth of germs and pathogens. So when something infects something, gets inside human body and it produces a illness, it is known as infection. Okay, so the invasion and growth of pathogen in a body is defined as infection. Now the germs or the pathogens can be bacteria, fungi, yeast or virus. Infection can be caused Infection can cause fever and other health problems or some symptoms depending on where it occurs in the body. Okay, then comes enzyme and protein. Now these are also related to each other. How? So enzyme, a substance that produced by a living organism which acts as a catalyst. Catalyst is one that increases the speed of reaction. Okay, which acts as a catalyst to bring about a specific chemical reaction is known as enzyme. Now enzymes can perform under different physiological conditions and there are different enzymes. Okay, so during an enzyme mediated reaction, a substrate physically attaches, you can see the substrate physically attaches to the enzyme at its active site, allowing the substrate to be converted to the new product. Okay, and then the enzyme is left as it is. Okay, enzyme does not get affected in a biological or biochemical reaction. Okay, it will just help to speed up the reaction. Okay, so you can see here these are these two substrates. This is the enzyme, it has the active sites. You can also see this works as a lock and key model. So both the substrates will fit properly to the enzyme and it will form enzyme substrate complex. Here the biochemical reaction will take place by using the substrate. Enzyme will produce a new product and then it will release the product and enzyme it is left untouched. Okay, now what is protein? So with the exception of a small group of catalytic RNA molecule, all the enzymes they are proteins, but not all proteins are enzymes. Okay, there is difference. All the enzymes are proteins, but not all proteins are enzymes. Okay, then all the enzymes are proteins which are made up of amino acids linked together by peptide bond. Okay, or peptide bond. So you will learn even protein formation and the structure of proteins in detail in your first year and second year. Okay. Now comes the vaccines and endotoxin. Now vaccine is what you are now familiar with, the coronavirus vaccine that we took recently. Okay, so vaccine is a biological preparation. That means it has a biological agent which is used in the preparation, and that provides active acquired immunity. Now acquired immunity is what? Something is injected to our body and it is outside, it is foreign to our body, so the immunity is generated to that, and this particular immunity is known as acquired immunity, okay, to a particular infection. So it contains an agent which resembles a disease causing microorganism, and it is often made by weakened or killed form of that particular microbe. That means it is not fully active to cause the infection, but it will just trigger the immunity. You will not get infected, but your immune system will recognize if you get an infection in further lifetime. Okay, so that's the role of vaccine. Now endotoxin. Now endotoxin is a type of pyrogen. Pyrogen is what? One that can cause fever. Okay, so endotoxin is a type of pyrogen and is a component of exterior cell wall of gram negative bacteria. Mostly the endotoxins, they are exterior exterior components to the cell wall of gram negative bacteria, and endotoxin is made up of polysaccharides. Okay, you will also learn this in, I guess, in your third year in details, but yes, you should know that what is endotoxin. Then preservation. Preservation is what you start learning, I guess, from your 12th, 11th or 12th grade in your biology. Same process is also included in your first year, where you study food spoilage and food preservation. So what is preservation? So it is a process of preventing the growth of bacteria and fungi such as yeast and other microorganisms, as well as retarding the oxidation of fats, which causes rancidity of food items. Okay, very simple. Just preventing the growth of microbes is what is preservation. Okay, then hyphae and mycelia. Now hyphae is plural, singular is hypha, and it is the filamentous thread of fungal mycelia. Now you can see the single thread-like structure is known as hyphae, and a network, a mat-like growth is known as mycelium. Okay, so mycelia is plural, singular is mycelium, and a network of fine thread-like structures is known as hyphae. Okay, network of fine thread-like structure called hyphae. Okay, network of all the fine structures which are known as hyphae is known as mycelium. Okay, then Burgess manual, very important to a microbiologist, is Burgess manual. It is the main source of determining identity of microbes and studying them. Okay, now there are two types of Burgess manual. Burgess manual of systematic bacteriology, it is used to classify unknown bacteria, and it is based on the 16S rRNA gene sequence. That means um, ribosomal RNA, 16 as part of a ribosomal RNA is sequenced, and it is studied for identification and classification of the bacteria. The detailed information about genera, species is mentioned, and methods for enrichment, isolation, information pertaining to the nomenclature is given. Here polyphasic approach is used. Now polyphasic is what? You study the morphology, that is the phenotype, the outer appearance of your bacteria, and even the genetic study is done, that is polyphasic. Okay, then comes the Burgess manual of determinative bacteriology. This is the second part, you can say, of Burgess manual, and it is used for identification and not classification and nomenclature. You just identify the given culture. Okay, this is what we actually deal with when we study our microbiology course. Okay, you are introduced to systematic bacteriology, but generally we don't use it for classification and nomenclature. We don't need to do that. We identify the cultures which we are given to. Okay, so it does not contain any descriptive information of genera and species. That means no information about enrichment and isolation of bacteria, but there are biochemical tests they are given. The other morphological characteristics they are mentioned, like you can say morphological characters such as cell wall oxygen requirement, motility, biochemical characters, everything is described, and depending on that we try to identify the given culture. Okay, so this was about some microbiological terms which I think first year students should know about, and I hope the concepts are clear here. So do like, share and subscribe to my channel, and do share these videos with your friends and recommend them my channel and tell them to subscribe, and thank you for watching.